Okay. Hi, everyone. I'm just gonna double check if someone on Zoom can let me know that they can still hear me. That's great. I moved the owl, so I wasn't sure.
Yep, you're good.
Hi. Today for the summer course, I'm going to be talking about specialized type of scanning that is used for fMRI. And the title, as you can see, from line scanning to tensor imaging, is essentially just a list of the things that I'm going to be talking about. A lot of people might not have heard of either one of these, so we'll kind of step through exactly what it means to actually do some of these things and what they're actually good for. Okay, so just right off the bat, we're gonna state that 2D and 3D imaging techniques, so almost everything. Okay, is it a matter of how close I am to the mic?
I think so.
Okay, I'll just move it then. I tend to turn away from the microphone a lot as well. Is this any better for people? Flash this. I'm gonna take that as well. Okay, we'll keep on. Okay, so most of what we've learned in the fMRI course so far has been about 2D and 3D applications. And this is kind of an easy way for us to think about MRI because it's the anatomical units that we typically think of MRI in, or anatomically. So you can either have 2D imaging, which is essentially just like a slice through a particular piece of brain, and then you try to image it over and over and over again, or you can try to do a volume, something like a 3-dimensional. That said, they're not the only tools that are available for doing neuroscience or clinical tests using MRI. I mean, this is kind of obvious, but the really, really expensive machine that we have downstairs, it can do a lot of things. It's not limited to just a couple. And so I'm just going to point out 2 examples today that we can talk about that can provide some extra information about certain types of experiments that we might want to do. And I'm just going to start with one of them, which is line scanning. Line scanning has been around for a very, very long time. It's been around, like most things in MRI, since the 90s. Although there were some experiments before then. But I personally think that one of the ones that really, or one of the experiments or papers that came out that really solidified why it's a really useful technique actually came out from NIH, and that's in 2014. Alan Koretsky's lab and Janu were working on some experiments in rodents where they actually tried to make the field of view for MRI as small as they possibly could in order to take advantage of certain resolutions that they can get by doing so. That's a little bit of a complicated way of doing this, but essentially the walkthrough is that they were imaging only this tiny slice of brain. And then if they turned off their phase encoding gradient and they just imaged very, very quickly along one segment of the MRI plane, then they could go both fast and they could go to really high resolutions. Now, this has a caveat that we'll go through, but at the same time, I'm just going to point out some of the really beautiful results that they had, which was essentially that they showed that depending on whether a stimulus should have been, a feed-forward type stimulus, so in other words, like telling us stuff about the sensory information that's coming into our brain, versus feedback, which is a longer connection type and potentially more internal processes, they can actually see through cortical layers here, labeled L1 through L6, that the patterns of the stimulus turning on, that response was different. And it was different in a way that made a lot of sense and a lot of people really excited, which is that in the feed-forward case, this is a stimulation paradigm, you're seeing activity first in L4. And then in the case where they were expecting feedback for the stimulus, you can see peaks starting at L23 and L5 first, followed by everything else. And this is on a time scale that's very different than we typically think of it. They're going very fast. So in this particular experiment, it was 15 micron and 15 milliseconds. Most of what I'm going to be talking about today is actually human scanning and how we can kind of develop some of these tools there. We can't achieve quite the same resolution, but we're trying. So even more just back to basics. What is line scanning? Line scanning is essentially a virtual electrode. It's a non-invasive way of basically sticking an electrode in someone's brain along the cortex and trying to measure different depths through the cortex. This is what I just said, non-invasively probing human cortical layers. And the goal is to scan at very high spatial resolutions through the cortical layers, but also at very high temporal resolutions. But it's also difficult. And I'll give you a couple of reasons why it can be difficult. Just like we saw previously in our little rods this is a pre-planning study, but basically this is the area of cortex that we want to image. What we normally see is more like planes or volume. So we would like to make it so that the excited volume that we're looking at is just that tiny little thing, but it happens to be that that's really difficult. You can ask me more about this in the questions, please do. But there are a couple different reasons why making pulses that are RF specific to only a small column is really, really hard. So normal slice selection is kind of what we're stuck with. And it gives us a good profile in one dimension, but it doesn't give us a great profile in other dimensions. It basically just gives us the entire brain. And as an example, we can see if you try to reduce the field of view of a particular slice of imaging, we would like for it to just reduce with whatever field of view we tell the scanner that we want it to look like. So we would like to say, okay, we're no longer doing 100, we want to do 50% of that. And we want it to just be that 50% in the middle. That doesn't happen. In MRI, we have an artifact, which is called a wrapping artifact. And it essentially means that all of that brain tissue that was in the 100% phase field of view is still there, and it has to appear somewhere. So it's just going to be wrapped around within our image. The most extreme case of this that you can possibly have is line scanning, because basically what we're doing is we're reducing our field of view, not by some percentage, but entirely. We're collapsing it all onto one line. For those that are acquainted with projection imaging, this is the same thing. But that's the problem that we're left with. So how do we deal with this? Well, we actually have to try to make it so that some amount of that middle of the image is the only part of the image that is actually excited at the time that we try to read it out. And the way that Alan Koretsky's group dealt with this, and it's also the way that I have in my own work, is to use what are called saturation pulses. Saturation pulses are essentially just pre-pulses before you start your TR that try to cancel out some amount of the signal that you have in that imaging technique. So this is really commonly used. It's not a really specialized technique. Saturation pulses are used for a lot of anatomical imaging in places that are hard to image, places like the spine or in body imaging, etc., where you want to really avoid something. But we're using it for a totally different purpose. This is essentially to cancel out all of the brain material, all of the brain tissue that we're not interested in. And so you can see here, in kind of the latest version of what we've been doing, we have 4 different saturation pulses in order to effectively cancel out all of that signal without also accidentally canceling out the part that we are interested in. And just to give you an idea of how this works, or what it looks like when it works, if we choose a particular line that we're interested in this case, we want to image motor cortex or somatosensory cortex. And so we're going to place kind of a single three-dimensional line going through that cortical sheet. Then, if we just look at what the line scanning profile looks like, bless you, we would normally see everything that's happening over this direction, the direction that we're not interested in. And this might have a profile that kind of looks like this, where you can kind of see a lot of brain in here. With the saturation pulses in place, you can essentially just leave a tiny bit of the image that's left in the middle, but cancel out everything else. Now we don't have nearly as big of a problem. Think about, okay, if the entire image is all collapsed into one line, it's not a big deal because it's only a tiny sliver of image anyway. Please stop me if anything in that doesn't make sense. But I think this might help if it's not coming together. So essentially what we saw here was what the line profile looked like in one dimension. Now I'm going to show you what it looks like in two dimensions, just to drive it home. So let's say we had this slice that we were choosing, and we wanted to make a line that goes through the image like this. We're interested in one, and we're also interested in well, we can place our saturation pulses in the correct place, and when we apply them, we're left with only an image that is just barely in the middle. For most purposes, this is kind of useless. It doesn't tell us a lot of information anatomically, etc. But at the same time, if you can then image this quickly and at high resolution, you do gain something only in this dimension. And that's the whole point of line scanning. That's our inserted electrode through the cortex that we didn't actually have to open the person's skull to. Here's just an overlay of what that looks like on top. It is up to the user, but at the same time, I would say the field is kind of latched on to a standard of about 3 millimeters. So you don't get much specificity along the cortex. Our aim is really to kind of leave that be, but try to go for high resolution through the cortex. But yeah, that's an area of
separation.
We absolutely do. We have a lot of limits for sure. And you'll see a little bit more. We also have some limits based just on that thickness. So for instance, if you're 3 millimeters wide and your cortex is curving through it, your voxels, which are, because of this profile, very thin through cortex, but very, very wide along cortex. If the layers are actually curving through them, that can cause some issues. But so we try to combat that. Okay. There's another way that you can actually do this other than using saturation pulses. I'm going to mention it briefly because it's really important, although I would like to see more development on it in the future. This is essentially a different type of sequence where you don't use any saturation pulses. You don't have to do anything before you start imaging, but you then use a refocusing pulse, so a typical spin echo type image, but you turn the spin echo plane so that it's orthogonal to the excitation. You can put it out on gradient, which is what the arrow indicates. And that makes it so that you can actually have your excitation and then refocus only the portion that was in the tiny little middle part. This is super cool. I think it's a totally awesome technique with one potential issue that's associated with it, which is that you don't get much signal out of it. We already know that spin echo doesn't produce quite as much signal as gradient echo style imaging. Here, that's kind of even worse. So basically what we can see on the top section in a paper from Mundo in Amsterdam is that the spin echo version which they call Selene, which is spin echo line, and has really, really great profiles in comparison to the gradient echo versions that they were using. They don't use 4 pulses, they use 2. But at the same time, the TSNR profiles that are associated with those are much, much, much lower. So just keep that in mind. I still encourage people to work on it if you're interested in acquisition question. Anyway, so what does it look like to actually have line scan data? Well, we went through this process. Now we're going to start recording our time series. The time series looks something like this. This is the profile direction. So in other words, every column of this little thing, that's one line. And we're just going to keep recording it over and over and over again. And we can do this pretty quickly. In some of the studies that we've worked on, we've gone up to 100 milliseconds. This particular case is about 250 milliseconds. But either way, much, much faster than what you typically think of with like EPI readouts. And then you can see kind of the cool thing is that on this one-dimensional little plot, we can actually mark what the areas that we're really caring about are. So I should have made this a little bit bigger, but on the left side, you can see M1 and S1 labeled. Those are the cortical ribbons that are associated with that little patch of cortex that we're looking at through time. Now, coming back to your question that had to do with profile, choosing profiles is really, really difficult in line scanning, and it takes a lot of pre-planning. It's not as easy as just kind of putting people in the scanner and just kind of going. That's partially because the patch of interest that you're normally looking at in experiments like this is really difficult to work with. You need to know a lot of information before you start. But secondly, you also need to know where you are in navigating on the MRI machine. So we did a lot of pre-planning in a lot of the studies that we have done. And essentially what you can see here is an example of choosing a position. Now, because the line width that you have is not perfect, it can be as wide as, like we said, 3 millimeters. We would often choose patches of cortex where there was no curvature. This is a completely flat little patch of cortex. And those would be the places that we would actually try to place the lines. And this just minimizes that effect that I was talking about, which is essentially just that you don't have kind of bleeding of different layers into different voxels of your signal. It tries to make them as independent as possible in each of the voxels. So what can it do? We've already gone through one example, which is the idea that you can actually look at time courses very early in responses at very high specificity in cortical layers. You can also do really interesting things with trying to understand what some of the mechanisms that are happening in different cortical layers when you're looking at longer responses. So this is an example that we've been working on recently, which is essentially a finger tapping response in humans, looking at those profiles that we saw, M1 and S1. And this is from a colleague of mine, Niels Mocknagel. He essentially, in the tapping that you see in the contralateral case, which is what we normally expect from a tapping experiment, you can see a really, really big fold response. And you can see here, basically just that. You can see with some specificity what's happening in the CSF. So these are things like vessels, veins, etc. You can also kind of see how it slowly goes down throughout cortex during the time of the stimulation that's happening. What's really interesting is that if you then go and look at the ipsilateral side, which is the case of the analog of the feedback that we're looking at, you can see something pretty cool, which is that you no longer see just a massive peak that's overwhelming the entire signal coming from outside of cortex. You can see dynamics from within cortex as well. And specifically, in motor cortex, that 5 is important you can actually see a peak that is happening there. And by peak, I mean it kind of stays the same while everything else drops, which is cool. Other things that you can look at are things like tuning. So because you have very fast scanning, you can potentially look at fast events, and you can try to get in a lot of stimuli in order to look at things like here we were looking at V1 tuning and essentially trying to understand whether receptive field size and orientation tuning, which are things of particular interest in a lot of the community in animal work, can be looked at in humans as well. And it turns out that, yes, there's some interesting stuff. Orientation tuning is less powerful in layer 4 than it is in the layers outside of that. So the infragranular and the supragranular. At the same time, PRF size, so receptive field size, how much of the visual field a particular patch of cortex is interested in, is minimal, kind of at the border between layer 4 and the supragranular layers. And that's a level of specificity that is really, really difficult to get in a lot of other types of layer images. Okay, so to wrap up a little bit with the explanations of line scanning, it's a specialized technique that is used for examining really high spatial and temporal resolutions, but only in one dimension. And it's useful specifically for looking at cortical layers. There are other ideas that people have had. When I used to work in Glasgow, people were interested in using line scanning for examining layers of skin as well in MRI, which I thought was a super cool idea. But for the most part, in fMRI, we think about things like cortical layers. Are there any questions about line scanning? Because I'm going to jump topics kind of one over.
Thank you. You mentioned the separation problem. So you have the what separation problem? What separation now behind that? Can you just make a multiband or just atom or something that one is not?
You can. Yep. The truth is there's the kind of steps of just getting it working in humans, and then there's the steps of optimizing things like that. I don't know everything that Nils is working on at the moment, but I would not be surprised if he's working on things like this. And particularly, I think ideas like using multiband are pretty awesome, just because, like you said, yes, it's more powerful pulse for saturation, but there are a lot of tricks that we know for kind of minimizing the power that's applied there. And if you could get rid of even just one of those pulses, it would be helpful. Because that's the major limitation in this particular type of scanning, how fast. If you could get it down to 2 pulses and then an RF and a readout, that's very easy to get within the Anything else? Oh, and maybe just one thing to add there. The reason for 4 pulses rather than just 2 is it's easy to think like, oh, you don't want to hit me, so you're just going to get everything out on that side and everything out on that side. That's easy. It's actually not quite as easy as that, because designing pulses to be both really, really sharp at their edges and also wide is difficult. And so the idea there was essentially to place 2 very sharp pulses that are not very wide, right at quarters, and then just blanket, get rid of everything else with some wider pulses that are less. Okay, now for something completely different. Tensors, what are they? Why do we care? In the simplest terms, tensors are mathematical generalizations of arrays, which I know are but specifically they're for cases where we don't already have names. So just to give you an idea of what this means, in terms of thinking about dimensionality, which is always fun, a 0-dimensional thing is a point. It's also a tensor, by the way. A one-dimensional thing is a line, and it's also a tensor. A two-dimensional thing is a matrix. It's also a tensor. And three-dimensional is a tensor. And everything past there is also that. So we can just think of that as like tensors are complicated data structures. That would be a totally okay way to think about it. How we get it into imaging is an open question, but yeah. However, I think there's maybe a little bit more useful way to think about it specific to neuroimaging, which is that any data structure can be considered a tensor, but there's something really, really useful to our particular work in this room, to thinking about the idea of coordinates being visible where we care about them, and less visible when we don't care about them at the moment. And what I mean by this is that when you open up your Nifty viewer, when you open up APNI, etc., very, very rarely is the first thing that you see not just the brain, because the brain is what we're all familiar with. That's the interesting part. And then often there are a few more clicks to get to, okay, if I click on this voxel within the brain, what is it doing? And what is it doing can mean a lot of different things. It can be a time series, it can be fusion directions, it can be all kinds of stuff. But we typically store our data in a structure that we understand visibly at first, and then we have hidden the things that are interesting about any given point that we care about in it. And that's oftentimes what we mean when we talk about things like tensor imaging. So it's super common. You've dealt with it before. This might seem like a little bit of a jump, but there's a reason that I'm going to talk about it here. And that's essentially that there's an inherent link between tensors and subsampling or undersampling. And the reason for this is that if we think about those tensors that we just talked about, let's say we have a picture of a brain. It's a three-dimensional brain. And then we have that for every single time point that we looked at. And we have it for every single echo that we looked at. This data structure is getting very, very large, very, very quickly, which is cool. Most of that's hidden to us, so we don't really think about it other than it takes up most of our computer frequency. But something that's really interesting about it is that MRI is time limited always. And in the other talk that I gave as part of the summer course, I talked about this quite a lot. MRI is very, very limited in how much data it can collect for every millisecond. You can try to push this hard, but you're never going to get past some inherent limits of the hardware. And so oftentimes we have to skip data. So you have this huge structure you want to fill in, and you would like to just spend the next week with the person in the scanner of filling in every single time point that you care about, every single echo, everything, blah, blah, blah. But we normally have to take shortcuts. One of the main ways that we take shortcuts is on things like filling in k-space. So we have to fill in the echoes and the time points faster. We have to skip some of the frequency information that gives us our spatial information. And here are a couple of different examples of what that looks like. There are different schemes and what the resulting artifacts that are associated with them are. But these methods typically take advantage of a couple different things that we talked about in the last portion, which is that there are correlations in our data somewhere. Oftentimes we think about this as correlations between the k-space points or between the channels, etc. But there are correlations in all natural signals. So the point of tensor imaging, which I'm getting to, is that you need to try to take advantage of those. And this is really interesting. I'm going to show one example of it, which I think is really, really cool and is an awesome method that we have been in discussions with PH about. trying to get them working on all of our scanners, but this is called EPTI. This is the equivalent of EPI, which we've all learned about in the summer course so far. EPI tries to cover the entirety of K-space after one single shot sometimes. And so basically you're just trying to collect as much data as you possibly can as quickly as you can. EPTI makes a little clever thing, which is that it counts time as one of its dimensions as well. Now you're not just trying to get through all of K-space, but you're also keeping track of the exact time after the RF that you actually collected those K-space points. And if you do this, it turns out that it has the advantage, which is that time is very, very well behaved after an RF pulse. So you can actually take advantage of the correlations that you know are there in order to reconstruct better. Super cool. So basically, the two examples that they have here are that we know what decay curves look like after the RF. So if you hit a particular place in K-space at, let's say, time 0 and time 20 milliseconds, and you can fit a curve between them, then you kind of know all of the points in between. So you took advantage of the correlations in order to fill in those data. And this is an important example of time resolve under sampling. So it's just another way for us to fill in more data, even though we didn't have the time to actually collect it when we wanted to. Okay. Hopefully everybody's on board, because this is another conceptual step forward. But undersampling can happen in any dimensions that we know have correlations. Decay curves make sense. We know mostly what they look like, so we can take advantage of that. But there are other processes that happen that are functionally relevant and really, really interesting to us that we can also take advantage of. So this is an example that I'm going to show from Tensor lab in California. And this is not brain imaging, but I think it applies to a lot of the things that we're really interested in brain imaging. So here, we're taking pictures of the heart. And we're taking pictures of the heart while it's moving, while we're doing different things to the RF pulses, things like allowing T1 relaxation, or like we just talked about, T2 star decay, etc. And if we keep really, really specific timing of when we collected every single K-space point, and we relate them to all the dimensions that we care about, we can actually try to resolve them using mathematics. So here is a little bit of a complicated scenario, but basically what they're saying is that we have these 5 pictures here. Those can all be put in a coordinate system that is described by all the processes that are happening. So for instance, this first one, the red one, it was imaged during a bright blood process from T1 recovery. So in other words, it was early in the T1 relaxation process. It was also at the end of diastole. It was at the end of systole. So it was at a particular process in the cardiac beat as well. Then we can move on to our next one. It's going to be slightly different. It's having relaxation that's happening to it. It's also changing the cardiac cycle, etc. But we can see that, okay, this is at a different point in our big data structure. So this is the end of diastole, and it also is still red blood. But then we start moving on. This one's also bright blood. This one's dark blood. This one's dark blood. So that's all during the relaxation time, but at the same time, we're also kind of moving around because of what's happening in the cardiac signal, etc. And basically, tensor imaging is just trying to keep really, really, really good timing records for every single data point that you ever collect, and then trying to model them so that they can be separated. So this is the goal of almost all of fMRI studies, is to essentially separate different processes so that we can tell what's happening that's due to something we did. But here, they are doing it in the case of chest imaging. And so you can kind of see that if they keep really, really good track of the cardiac signal, the respiratory signal, the T1 recovery, and the T2 prep that they're also doing, all these processes can kind of be separately looked at, which is pretty cool in my mind. I wish it would continuously play. So here you can see that just the cardiac is moving. Here, the lungs are moving, but the cardiac is really not, etc. So it's cool. The specific models that are used are basically up to whoever's designing a specific reconstruction algorithm here. But in the case that we just looked at, they were using what's called a low rank tensor structure, which is essentially just that you can do something like PCA, but rather than having only 2 dimensions like you do in typical PCA, you now have many dimensions. This is a class of mathematical problems called tensor decomposition. And they are awesome. So I'm going to give an example that has been something we've been working on here at NIH, and some of these things are actually available to play with a little bit. Tensor imaging is something that we just had to walk through basically what it was. It's a little bit easier to call it something that we're actually really, really interested in, which is that we want not just time resolved experiments, but we want functionally time resolved experiments. And so basically what this is doing is it's trying to keep track of all the same things that we were just talking about, but we're adding in things like trial time, trial response, trial type, etc. So this is actually a way of looking at your data so that you can try to build whatever analyses that you would do, things like a GLM, into the reconstruction process so that the images you get out are actually the ones that you're really, they're the final product, they're the things you're interested in. And the reason that we can do this is, again, because as we're acquiring case-based data, we can think about all the things that are happening simultaneously, and we can just try to keep track of them. Things like the experiment, but also breathing and heart rate, because there are particular types of responses that you can imagine are different depending on if they're out breath or in breath, etc. To dive a little bit deeper into what this means in terms of reconstruction, we're just going to talk about a typical EPI experiment where we try to cover as much of case-based coordinates, in case you're wondering. We're going to try to cover as much of case-based as we possibly can, and then we're going to reconstruct it. The little boxes describe an image reconstruction. And we're just going to repeat that over and over and over again until we end up with a time series. And then we also hopefully have a log file and some data from our biopack and all of these things. And we can try to take care of these after you've already done the reconstruction. This is what we typically do when we do things like fMRI denoising and GLMs, etc. But you can also think about this during the reconstruction process, like I said. So basically, we can just continuously go through case-based at whatever time we want, whatever's best for the imaging modality that we really want to get at. And we can kind of just overlay all of our trial structures that we were interested in, and they're just kind of wrapping around this structure now. So we're now no longer keeping track of things chronologically in comparison to the beginning of the run. We're keeping track of them in reference to the thing that we were interested in. So this is the trials things. And then we can fill in as much of case-based as we possibly can, and we can try to reconstruct the time points that mattered to us in whatever experiment it was that we cared about. So here, let's say, rather than these lines being a little bit slanted because it took time to get those images, we can just take all the things that are really close to this time point that we want to reconstruct, and we can try to reconstruct it. We have a time course of that particular trial response, etc. Again, we're going to use some mathematical tricks because we couldn't fill in that entire thing like I showed it in the little diagram, but there are missing data and you need to figure out how to fill it in. But the nice thing about it is that this means that you can use a lot of the techniques in tensor imaging that you would not normally think about in functional imaging. You would think about it more like anatomical imaging, which is that you just go doing whatever it is that you need to do, and then you eventually get to the point where you do your reconstruction. So, for instance, this is an example of data that we collected downstairs in the NMR center. This is essentially just a very, very high resolution functional scan, but the images themselves, because there's so much data that's kind of surrounding it, can be analyzed like they're anatomical as well. And on top of these things, you can do whatever it is that you need to do. That's also important for your studies. So in this particular case, we also recorded multiple echoes. And this makes it so that we can actually get out parameter maps of the anatomical information for proton density as well as T2 star amounts. And then we can analyze those. We'll start with just this one. Basically, well, kind of just this one. So we can basically look at our cortical layers here on a segmentation of our functional data. And then we can look at the parameters that are associated with the cortical layers. So here we're basically just plotting all of the voxels along the cortical depth. And then we're measuring what the T2 star values and what the S0 values do throughout this cortical. So in the T2 star case, this is a V1 process, by the way. This is important because we have this T2 star dip in the middle. And that indicates that we actually know a little bit about where one of the particular layers is, which is the layer 4, which is associated with calcarine and the striatum, is kind of in the middle here. And we have a pretty good location for that. At the same time, you also get a zero value, so you can kind of see that the proton density is largely flat during the gray matter, and then it's going to kind of shoot up as we get outside of it. More importantly, we can then take our data and also arrange them in the order that they were collected in comparison to all of the checkerboards that we showed, which was the little experiment that I showed a few slides ago. And we can just see what the response looks like. How does the image actually change as a result of that process happening? And it turns out that if you look at these same cortical layers, we can see a response to a relatively short stimulus here. And what's more is that we have a very sharp response here, and we have a very long undershoot, which is already pretty interesting. But more importantly, we also have a higher response in only the upper layers as compared to the other ones, and you can see some really differences as well. So those are all interesting things. This is not an analysis that is particularly easy to do without upsampling, etc., in other layer studies looking at Lorenzo. Anyway, the point being that those are the things that I was interested in when I was reporting those experiments. They don't have to be what you're interested in. So I can give all kinds of different examples of the different dimensions that you might care about a lot and that you might record along with your fMRI data and want to reconstruct. Things like, I didn't show anything about respiration or cardiac. Those are really, really interesting processes in the brain that we might want to reconstruct. We might want to see things like T2 star or T1 or modulations in flip angle. It's an excitement in the field at the moment. Or you might really care about something about your stimuli. Things like, oh, what were the spatial frequency or the orientation processes that were happening at any given time, etc. So I think I've covered this pretty well, but fMRI relies very heavily on 2D and 3D methods, as it should. They're really, really awesome, and they work really, really well. But there are some cases where detailed scientific questions might call for really, really specialized types of imaging. And these are a couple of the tools that we talked about today that are interesting for doing very, very specific experiments. You would never choose to do line scanning if you cared about network processes that were happening across the brain. You can't, it won't give you that. But it can give you some very, very detailed information about specific experiments that you might want to do. And the same goes for FTR. It's a more generalized case, but at the same time, tensor imaging has capabilities to actually resolve things that not all imaging types are capable, or at least not in this case. So I'll leave you with that. Thank you very much. If there are any questions, please